integrating computational drug design, biophysics, and population genetics to prevent the emergence of resistance. Eugene, the floor is yours.
Okay, thank you very much, Michael. And it's great to be here, though, virtually. And I put even a little kind of a small, my favorite place in Paris. Kind of, I miss real Paris, but unfortunately, that's life. So kind of, but still, it's great to be here. And I'm very excited to present our recent and not so recent work, which is sort of along the themes of this conference. And of course, what I show here is something that is a fitness landscape, which is sort of a metaphorical description of distribution of fitness effects, or whatever you can call it. And of course, it shows some schematic kind of aspects of epistasis, schematic aspects of evolutionary dynamics, but kind of in many cases, if not most cases, it's mostly a conceptual diagram, because we know pretty well, or to some extent, kind of what is on the vertical axis. We can define fitness in some reasonable way through some operationalistic terms or through some kind of sort of experimental reality. But what is on the horizontal axis, kind of very often, kind of it becomes a little bit murky, because we don't know what are the underlying physical chemical processes that determine fitness or determine fitness effects of mutations. And to that extent, it is still somewhat a conceptual diagram. And what we kind of were trying to do over the last maybe 10 years or 7 years, whatever, joining in my lab computational, both computational and experimental effort, is to try to map the axis on this fitness landscape, horizontal axis, and to try to understand kind of a little bit better what are the underlying traits, which are kind of microscopic traits, molecular traits, which determine fitness effects of mutations. And then, of course, to apply this understanding, if any emerges from this type of studies, to kind of to more informed evolutionary models, including kind of models where real evolution occurs kind of on medically relevant time scales, including evolution of antibiotic resistance of antiviral, or evolution of viruses escaping antibodies and things like that. But try to use as an input a DFE, or fitness landscape, which is more informed of macroscopic scenarios, and specifically what kind of mutations give rise to what kind of fitness effect, so that kind of ideally to predict and to play some sort of chess with pathogens to predict what the next mutation might be, and to try to proactively design some stressors or some antibiotics, if it's bacterial scenario, to be able to suppress this resistance. That's of course a tall order, and that's a difficult proposition, but I will try to show that there are certain steps that can be made kind of along this route. And now kind of the experimental approach, how we try to address this experimentally, is shown in this diagram, where kind of we combine computation kind of as a first step, trying to determine what kind of variation we would like to design, and kind of to introduce the bottom-up variation, genetic variation through genomic editing into specific genes of interest, which have some physiological significance, or which are drug targets, or whatever, which are involved in some evolutionary process, and then create, in this case, bacterial strains, but we also work with yeast, which carry specific kind of mutations or more broad variation in their gene. Rationally, we know what kind of variation we introduce, and simultaneously to determine biophysical effects of this variation, to purify proteins which carry this mutation, and determine a bunch of their biophysical properties, and then try to link kind of biophysical variation at the molecular level to fitness variation at the organismal level, and to see whether there is kind of a link which can be described as the more smooth fitness landscape rather than purely probabilistic, but to present a fitness landscape which is more kind of biophysically informed, predictable, and then can be again used as an input in evolutionary simulation and population genetics analysis, in order to see what kind of evolutionary process can entail. So this is kind of the idea, and there are several papers which are published from the lab, which highlight how this is done. Kind of there are technicalities, of course, there. But the problem is the question that we encountered when we first started by introducing point mutations into a specific gene, which I will talk about, DHFR, is that point mutations usually provide relatively limited variation of biophysical properties. So the dynamic range of variation which you want to map to fitness is relatively small. So point mutations are not a good vehicle for this type of investigation. And so essentially kind of we work, as I said, with this enzyme DHFR, which is a core metabolic enzyme, and which kind of function is very well known, and it is implicated in synthesis of nucleotides, some amino acids, etc., etc. It is essential enzyme, which is very important. When you kill it and when you knock it out, the cells, E. coli cells, cannot grow. So unless there are some certain supplements in the media, which comes handy under certain conditions. And so kind of focusing on this enzyme, but this is not the only one that we worked with. For example, another one is ADK, which Michael Manhart, who is here, who was a postdoc in the lab, kind of contributed quite a bit to. But today I will focus on DHFR. And as I said, kind of the initial attempts were, and they were sort of successful, is to introduce various point mutations informed by molecular simulations with kind of in a strategic parts, and to see what are the molecular and fitness effects of these point mutations. But again, the limitation is that these point mutations are of relatively limited dynamic range in terms of how they change biophysical properties of the protein. So we selected an alternative strategy. And the strategy for that is to, instead of kind of introducing point mutations in the wild type E. coli DHFR, we kind of looked at DHFRs from different sources, like here is shown for the genetic tree for DHFR and the distribution of sequence identity with E. coli. And instead of kind of introducing point mutations into wild type DHFR, we completely transplanted DHFRs from different organisms into E. coli background, into E. coli genome, in the hope that the variation of biophysical properties of these various DHFRs, which have various degrees of divergence from wild type E. coli, will allow us to have a sort of a broad variation, which will allow to map a fitness landscape more precisely and more accurately. So that is the idea. And of course, it requires not only kind of creating this transgenic E. coli, which carry DHFRs from other organisms in their genomes, but also purifying these DHFRs from these different organisms, and again measuring their molecular properties. But that has been done with 43 DHFRs of different degree of divergence from E. coli. So we did 43 strains, and this is the work mostly of Shimon Bernstein, who is now professor at Ben Gurion University in Israel. But when he was a postdoc in the lab, he did that. And so essentially we have this zoo of this transgenic E. coli, which have these 43 different DHFRs in their genomes instead replacing wild type DHFR, and also the zoo of these 43 DHFRs, which were purified and measured, whose biophysics was measured. And so this is kind of the idea, and that's what we found. We found a broad distribution of growth rates for this transgenic kind of transgenic E. coli's. And some of them grow faster than wild type E. coli. Many of them, most of them grow slow. Some of them could not grow, despite the fact that these were active, fairly active DHFRs. And this is kind of shows the distribution of their molecular properties, kind of stability-wise. Some of them are more stable. They melt at higher temperature than E. coli. Activity-wise, there are quite a few which are much more active than E. coli, like almost order of magnitude in terms of catalytic activity, more active than E. coli DHFRs. And also very important part of this kind of equation is intracellular abundance, how many molecules exist in E. coli cells, kind of which we had to measure. And again, some of them have a very low, this is logarithmic scale, have a very low abundance. Some of them have similar to E. coli, and some of them much greater than E. coli abundance. And the prediction kind of what it was done with the aim, kind of the underlying hypothesis in a sense for this experiment, that we will see kind of the distribution of the fitness effects upon this variation, how this growth rates depend upon variation, which can be sort of predicted and measured in the terms of molecular properties of transplanted DHFRs, kind of so that it is catalytic activity, stability, and the total abundance. And this was done initially kind of in this paper, and we kind of worked a little bit extending this model to make it closer to prediction. And so that is kind of the prediction. And the idea is that the fitness, in this case growth rate of bacteria, is proportional to the flux through the enzymatic chain. In this case, it's a folate pathway, because this folate reductase is a part of the folate pathway, which is one carbon donor for many processes in the cell. And so the flux through this folate pathway, which is about 11 enzymes, kind of, it is constitutes fitness, is proportional to fitness, the growth rate. And so this is the prediction, and kind of this is the curve that is predicted, which shows that at high activity of the enzyme in question, it becomes a plateau, because the rate limiting step are enzymes which are other than DHFR in this chain. And so this is kind of the predicted behavior. And of course, the big question is whether this behavior is observed. So now with this 43 transgenic E. coli, we considered kind of various correlations between fitness or growth rate, which we measured in different molecular properties. And kind of this is again is published a while ago, so I won't go into detail. But what we have found essentially is that indeed this flux prediction works really very well. So when we plot feed growth rate kind of for the E. coli, which can be for the DHFRs, which can be expressed on the E. coli background, some of them cannot, and we understand now and know why. I don't have time to discuss, but for the ones that express in E. coli, kind of the foreign DHFRs that express in E. coli, we see a pretty good kind of consistency with this flux balance theory prediction. And there is one outlier, but here for this outlier, there is another mutation on the background, which is unrelated to DHFR, which we determined from whole genome sequencing. So we understand why this outlier appeared. So now kind of the similar behavior was found for this enzyme, which we also worked the ADK, and this is published in this paper in 2017. So if you're interested, you can look at further. Now, what we kind of try to do is to apply some of these ideas to try to understand kind of very interesting evolutionary problem or evolutionary scenario, because DHFR is a drug target. There is a trimethoprim, the compound which targets DHFR, inhibits DHFR. It's an antibiotic. It's not. It's a part of antibiotic bacterium. There are two antibiotics in the bacterium formulation, and so when you were children, probably you took that kind of early in your life. And so one of the components of bacterium is trimethoprim, which is a DHFR inhibitor. And in this work from my colleagues at Harvard, Roy Kishoni and Dan Hartle, kind of they studied the progression of emergence of resistance against trimethoprim in E. coli. So they use this device, which I'm sure all know about, morbidostat. And then they did sequencing of the FOL8 gene, which encodes DHFR. And then they asked what kind of mutations arise in response to this kind of trimethoprim treatment in this continuous morbidostat evolution, and then outlined kind of a series of mutants in the FOL8 gene in open reading frame and in actual gene, which kind of constituted steps towards resistance, and they were outlined in this paper. So what we tried to do kind of as a sort of follow-up to this study is try to make sense of these mutations and of the order in which these mutations appear, trying to understand whether we can predict the order in which they appear, whether there is some rationale into that. And for that, of course, we need to know what these mutations do to DHFR, why are they resistant, what kind of factors change in these mutants, why they resist trimethoprim. And to that end, kind of what we did, we created 8 strains of E. coli, which carry each of these mutations. And combination, there are 3 mutations in open single mutations, there are 3 double mutants, and then there's 1 triple mutant. And so we made these strains, each carrying this particular variant of DHFR, measured its IC50, and then kind of what we did, we for each of these strains, for each of these variants of DHFR, we measured their various molecular and cellular properties. This is published here, so I don't have time to go into great detail. But the point is that essentially kind of the question was, can we predict fitness from these measurements of molecular and cellular properties? So if we know exactly what this mutation does to the protein, can we predict what this mutation does to the organism, what will be fitness here? Or IC50 concentration of antibiotic when growth rates falls to 4. And these are these IC50s, kind of the degree of resistance to antibiotic, which are shown for different variants. The most resistant variant is a single mutant L28R. Again, this is the concentration of trimethoprim, which is required to drop growth by half. This is kind of the definition of IC50. And so the greater IC50 is, the more resistant the strain is. And so these are the numbers. And wild type IC50 is much, much smaller, meaning that it is not resistant. And then again, we kind of tried to predict this IC50 from purely molecular properties of DHFR. And again, kind of this is, I don't have time to go into great detail, but the point is that if we use this kind of flux as a proxy for fitness, and then we can kind of relate flux with individual molecular properties of DHFR, we can indeed see that for many, many variants with trimethoprim, without trimethoprim, etc., this is the flux balance theory prediction, metabolic kind of balance theory prediction. And this is kind of the data, and you see that they follow kind of reasonably well the data. And this is kind of the equation for fitness prediction for fitness from metabolic flux balance theory, and that then you get. And then kind of you can use this flux balance theory to predict IC50 and compare kind of this prediction IC50, the resistance to antibiotic phenotype, with experimental IC50. And as you can see, the prediction is fairly good. And this is kind of spans many orders of magnitude, because the different mutants have different resistance, kind of like about 3 orders of magnitude in IC50. And this is kind of the prediction when we, instead of using actual measurement of concentration, we use the prediction based on kind of theory of dynamics in intracellular dynamics, which I don't have time to discuss. And interestingly, kind of there is definitely a trade-off between escape of drug inhibition, meaning that it doesn't bind trimethoprim as strongly, and catalytic activity. So if it wants to escape inhibition, it has to sacrifice catalytic activity. So now kind of we tried to make a step further and to use kind of population genetics, quasi-species with noise simulations, to predict the succession of mutants which are now known. So this kind of plot shows how different of this, each of these variants grow in the presence of different amounts of trimethoprim. And as you can see, wild type is the best at 0 trimethoprim, but then different strains at kind of at various concentration of trimethoprim take over. And there is one strain which is really kind of this, it shines, and this is a double mutant, one of the double mutants. And so basically what we tried to do, we tried to kind of to do evolutionary simulations to predict the succession of mutations, and then design small molecules which will proactively kill some of the most dangerous kind of, I would say, mutations here. And kind of basically what we found, that there is a particular mutation which is really very dangerous, because when it arises, there are different kind of paths to resistance appear. And the reason is that this is the only variant which is more stable than the wild type. So it epistatically provides reservoir of stability, which epistatically allows other mutations then to emerge and fix. Kind of the next step was to design these small molecules, alternative to trimethoprim, against this variant, L28R and others. And there is kind of, I have a subgroup in my lab does some drug design. We developed this algorithm, which is now almost 25 years old, which allows to design small molecules for given variants. And it was kind of done, there was a workflow, computational. And so at the end, kind of we came up with several compounds and new compounds. And these compounds indeed showed uniform inhibition, not only of wild type variant, but also this kind of mutant variants. And that, of course, was kind of quite promising in the sense that that's probably they will inhibit resistant variants. And indeed, kind of what we found is that these compounds, this is one of the most interesting compounds, it indeed kind of inhibited this L28R variant. Now it is growth. This is in vivo data, how that inhibits the actual growth. And as you can see, that with trimethoprim, the IC50 is huge, meaning that it is resistant to trimethoprim, but to this variant, this mutant cannot resist this compound, this variant L28R, kind of the compound which we designed is effective against. And then kind of that proof is in the pudding, and what we had to do is to show that it does not evolve the resistance, that this new compound does not suppresses the resistance, and we have this kind of very nice evolutionary robot, and there is algorithm which was developed by João Rodrigues, which kind of runs evolution at IC50. So when the new mutation arises, the robot automatically increases the concentration of the stressor. And as you can see, kind of to 3 meter prim, it quickly evolved the resistance, which is about 1,000 times greater IC50 than originally. I'm almost done. And but to this new compound, this kind of IC50 did not arose a little bit by a factor of 2. So the wild type, the IC50, the resistance to this new compound for the wild type is this 70 micromolar, and to the kind of the evolved strains, the resistance is about 130 micromolar. So there is a weak resistance, but nothing close to 3 meter prim, kind of, which is a factor of 1,000 increase of IC50. And then kind of we did whole genome sequencing of the evolved strains under the CD15-3, and what we found, this new compound, and what we found that the only way for the bacteria to evolve at least weak resistance is to duplicate part of the genome which is responsible for efflux pumps. And so what it did, kind of, it increases the efflux pump activity, and that pumped out some of this compound, but not sufficiently to make it truly resistant. Okay, so I'm out of time, and I want to just summarize. And kind of the key point which I wanted to really kind of a take-home message is that kind of when you try to fill this concept of fitness landscape with some molecular detail, which we call biophysical fitness landscape, you really can get some predictive models which can be used further to kind of to proactively, for example, design stressors or do kind of things which could be of interest in biomedical application and also in evolutionary applications. And that gives you a very detailed fitness landscape on which you can test evolutionary theories by doing simulations and comparing them for different kind of time scale experiments. And then with whole genome sequencing, you can understand also what kind of epistasis can be around and so on. And that's actually being done and is done. Okay, so this is kind of something that I didn't talk about, but the supplement to this analysis is a very powerful tool, which is metabolomics, which allows really to understand things way beyond the target gene and kind of how things change in the cell. And that's something which is becoming more and more kind of common to this type of studies. And finally, but last but not least, these are the people who did the work. Kind of Shimon started experimental work in the lab 10 years ago, and Joao is still in the lab, and Surav, they were working on evolution drugs. And these people who are now kind of professors elsewhere, they did theoretical work, population genetic studies, etc. And we also collaborated with my great colleague Dan Hartl and Elena. Thank you.
Thank you, Eugene, for a great talk. We have a question from Martin White concerning the.
Sorry? Maybe I can say it because